Okay, thanks, Stacy. Can everybody see the slides now? This is a beautiful one. This is actually Syntrichia ruralis. This is up in the Sierra foothills. It has a lot narrower leaves than Princeps does, and they're more squarose recurved. Once you get good at it, you can tell the ruralis from the Princeps. They're both in the same environments, ruralis in a little bit more xeric environments. So you've seen a bunch of the traits already mentioned as we were talking about variation among the different species. If you were to look at a taxonomic treatment, which this is one, a description of a new species, the kind of characters we're looking at are the leaf characteristics like shape, the length of the hair point. We're looking at anatomical characters of how the leaves look. A very important set of characters is in the cross section of the midrib. So this is a cross section of the midrib about here, and the presence or absence of different kind of cells, including the conducting tissue, which is here and here, have to do with taxonomy. Here's one of them that has a conducting tissue in the middle of the stem, pretty much vestigial. The water movement is as we were talking about before, but these guys do have conducting tissue. Many of them do. So these are the kind of characters. Here are those big window cells. Here are the upper cells. These are to the same scale. The length of these basal cells is a really important character. The size of them in general. You can tell Princeps from ruralis just on the size of these basal cells. So these are the kind of taxonomic characters. We don't have time to do actual taxonomy and keying out of Syntrichias, but if we did, these are the kind of characters that we'd be looking at. We'd be making lots of cross sections. Here's a slide that one of our collaborators, Mandy Slate, put together. This shows the North American Syntrichias, almost all of them that we're looking at. Can point out a few of the ones we've been talking about today. Here's Canonervis. Very ovate leaf, relatively short hair point as compared to some. The one that we were working on together was Princeps. Where's Princeps in here? Here. Can't really see its hair point, but it's fairly long. Some of them have this panduriform shape. That's another key character we use, like Obtusissima here, where it looks like it has a waist on it or a narrow place like on a violin. Panduriform means violin shaped. Whereas others of them have pretty much parallel sides and don't have that indentation. The relative length of the group of basal cells is another taxonomic character, and so forth. That just gives you some idea of how these are roughly to the same scale. So it gives you some idea of the variation of size and of the traits. As I mentioned, some of them don't have hair points at all. So in terms of ecology, they vary a lot. Here's some of our collaborators out in the desert west of Las Vegas. That's me, Lloyd Stark, Nick McCletchy, and John Brinda. We've seen lots of pictures of the Mojave type desert situation. This one is actually not Canonervis. This would be ruralis. You can tell because it has the leaves kind of twisted around the stem, a little narrower. But even though we've emphasized the really dryland ones, there are species of Syntrichia that occur in much more mesic habitats, never in wetlands, but in woodlands, mid-elevation woodlands. And even when you get up higher, Syntrichia norvegica is a fairly common moss in the subalpine and alpine of California and other Western areas. So you go all the way from alpine down to the really dry deserts where we've been looking at Canonervis. I showed this slide before, but this just underscores the functional significance of some of these characters. Javi was talking about these window cells at the bottom of the leaf. And as he pointed out, and we were discussing it, at least in my breakout room, these have perforations. The cell at the end of its development actually resorbs away its cell wall on the outside, and there's a pore. So you can actually, if you were a little bug, you could walk from this side of the leaf to the other side of the leaf through that pore. And that's the route that the water takes when it's coming from the bottom of the stem up through. You see how sheathing these leaf bases are, but these cells are standing open. So water can go up this tomentum, go right through the leaf to the other side of the leaf and keep on going up the stem. And then we all observed with our demo the movement of the water by capillary action through the channels that are created by these papillae. The leaf is only one cell thick. The papillae are hollow, and it appears that the function of the papillae are actually to serve as little snorkels. They kind of look like snorkels here. When a plant is completely immersed in water, it tends to slow its photosynthesis down because CO2 moves through water much more slowly, about 10,000 times more slowly than it does through air. So it's to the plant's advantage to have one of the surfaces kind of out in the air. And when these mosses have reached their turgid full state where they're hydrated, and then if you look at them closely, you'll see little papillae tops sticking out like little islands. And the chloroplast, this is a TEM view of a cross section. Here's a papilla, and here's a chloroplast that's sticking up into the papilla. So the goals of this part of the project were to look at the systematics. We want to try to figure out how many species there are, roughly. We want to try to figure out how they're related to each other. And then we want to reconstruct some of these important ecological changes, biogeographic changes. How has it moved around on the Earth's surface? How has it gone from one kind of environment to another? We wanted to actually figure out what Syntrichia is. Syntrichia is not necessarily clearly understood, and people have different definitions of it. So we want to know what it is and what its relatives are. What are the major clades, which is a name for a lineage? What are the major groups within Syntrichia? What's its biogeography? Can we infer where it might have originated and where it might have moved to? And then looking at this, all this wonderful array of morphological characters, how has that evolved over time? So we took a, this is a approach, it's called genome skimming. And what it does is it's not nearly as careful of sequencing as you would do when you're going to reconstruct the whole genome, as Mel Oliver talked about, but it's like it. What you're doing is sequencing bits of the genome to a shallower depth and putting those together. So think of it as a very sketchy genome that you're getting, but it's less expensive to just kind of sketch out the genome than it is to try to really assemble the chromosome level quality that Mel was talking about for our high quality genomes. We had 608 samples that we collected. Many were herbarium specimens, some were from what we had collected. We tried to represent all the things that had been called species. Many of them just appear not to be related to each other, but we at least used them as a factor to stratify. And then we built a lot of trees. And I can't show you all of them. We've looked at the genes separately and then the genes together in combination to try to figure out what's going on. So I thought I would just show you an outline. Someone asked in the chat the difference between Tortula and Syntrichia, and I'm going to now get into that. I want to first show you the overall topology is like this. What we're considering to be Syntrichia is a group, a clade. This is a monophyletic group. We're going to call it about here. These groups that are outside of that, so these are the closest relatives of this clade that would be called Syntrichia. These are mostly Tortulas, although there are several other genera in there too. Genera you might know of like Padia or Henadiella. The group that has been called Tortula, so Tortula has been used as a very inclusive taxonomic term that could have included most of what we're looking at here, if you had applied the name here. if you separate out Syntrichia and you separate out these other genera, you end up with a tortula that is a wastebasket that really doesn't have a nice definition. There is a group of tortulas that I would restrict the name to that does include the type that some of you might know, Tortula inermis, which is also common in deserts, and Subulata, Muralis, Brevipes. These are all common California. Syntrichias, they're this group that's fairly close to Syntrichia. And then you have other tortulas out here that have been variously put in the genus Podia, and others. So the taxonomy further out from Syntrichia is still going to need some more work. We're not working on the whole family. We're just trying to sort out what Syntrichia is and at least what its closest relatives are. Within Syntrichia, there's lots of interesting things that one could talk about. In blue here are names of mostly species, but also groups of species. In this tree, Syntrichia princeps branches really distinctly from many of the others. We have in these clades here, we have mostly the Syntrichias that like to grow on rocks or trees. This is kind of the rock tree clade here. We've seen Papilosa, I showed you the one with a papillae on it, and Pagorum with the leaf-like, with the little gemmy. Remember those little round gemmy I showed in that one picture? And Pagorum had the leaf-like gemmy. So we have a bunch of rock and tree Syntrichias. The groups that Javi is studying most intensively, there are a number of separate lineages that are in South America. And then probably the most interesting thing that we discovered in the phylogeny part of this is there's a North American hemisphere clade here, which is a monophyletic group. It includes Canonervis, which we've been talking about a lot, and it includes Ruralis, which we've been talking about a lot, and a whole bunch of other potential segregates that have been pulled out. I mentioned Norvegica, which is the alpine one, and subalpine one. It comes out as a little group, but it's nested inside of a bunch of things that are called Ruralis. Somebody was asking about Syntrichias on sand. There's a form called Ruraliformis, which grows on sand, which does not come out as a group here. It may be just a phenotypic modification because it occurs several places here. The thing to call your attention to, though, these branches are scaled to the number of mutations that are reconstructed to have occurred on them. And you can get a rough idea of the age of a group, very rough, by looking at the number of changes that have had time to accumulate. And the thing that was amazing to us is that the whole group of all these Ruralis complex things and Canonervis look to be a tightly related group, incredibly different morphologically and physiologically and ecologically. And they appear to have radiated. You've heard the term adaptive radiation, which is when you have a bunch of lineages arise in a very short period of time in evolution. We think we might have discovered one of the few examples of adaptive radiation that have been documented in mosses, where we have some incredible amount of morphological, physiological variation that has occurred approximately, 5 million years or so ago, depending on how you want to calibrate this. But it's roughly about the time that a lot of the other, it's when the dry lands evolved in the Northern Hemisphere, and you had groups like Ceanothus and Arctostaphylos radiating and things. This might be something that's gone right along with them. And the reconstruction for the biogeography would be this was a single dispersal to the Northern Hemisphere from Southern Hemisphere groups, possibly by an alpine sort of hopping route. They're on the high volcanoes coming up through Central America. There are Syntrichias of the South American type up on those. So we're just kind of, looking at this and trying to reconstruct what we're doing with this. But this just shows you some of the traits that we think have evolved in the different groups. And then one more slide is when you have a phylogeny, you can start to map on traits and biogeography and try to understand what's happened in evolution. Here's a southern hemisphere, northern hemisphere comparison. There's a good chance that the whole group evolved in the southern hemisphere, and then the incredible diversity in the northern hemisphere is a result of a few movements out of the southern hemisphere, is the way we're leaning right now. We're working intensively on getting more data. This is a very hard thing to resolve right here. Then a few traits. We talked about asexual reproduction is not randomly distributed on this tree. There appear to be certain clades. These are the ones with dark green here. This is the pagorum here and the papilosa here. That whole ruralis complex, the whole northern hemisphere radiation, doesn't have any kind of official asexual reproduction. Propagules, even though it does reproduce asexually. Hair point, dark red is where you have a hair point, light red is where you don't have a hair point. So those are just examples of how once you have the phylogeny, you could start to reconstruct the order of evolution of the traits that are interesting.